Hello, welcome to the Sleepedia channel. This video presents a scientific explanation of what the human brain does during sleep, focusing on neural activity, memory, and consciousness. Sleep is a universal biological process observed across nearly all complex forms of life. Despite its apparent passivity, sleep represents one of the most active and essential states of the human brain. For much of scientific history, sleep was considered a period of neural rest, a temporary suspension of cognitive activity required solely for physical recovery. Modern neuroscience has fundamentally revised this view. Sleep is now understood as a highly organized, metabolically active, and neurologically indispensable process during which the brain performs functions that cannot occur during wakefulness. From a neurobiological perspective, sleep is not defined by inactivity, but by a systematic reconfiguration of brain activity. While behavioral responsiveness decreases and voluntary muscle activity is suppressed, large-scale neural networks remain active and, in some cases, display patterns of activity that exceed those observed during wakefulness. These changes are not random. They follow a predictable architecture governed by intrinsic neural rhythms, neurochemical modulation, and homeostatic regulation. Sleep serves multiple critical functions, including memory consolidation, emotional regulation, synaptic reorganization, metabolic waste clearance, and the maintenance of neural plasticity. The failure to engage in sufficient sleep disrupts these processes, leading to measurable impairments in cognition, emotional stability, and long-term neurological health. Understanding what the brain does during sleep, therefore, requires examining sleep not as a single state, but as a sequence of precisely structured neural phases. Human sleep is organized into recurring cycles, each consisting of two fundamentally different brain states: non-rapid eye movement sleep, commonly referred to as non-REM sleep, and rapid eye movement sleep, known as REM sleep. Across a typical night, the brain transitions through these states approximately 4 to 6 times, with each cycle lasting between 90 and 110 minutes. The relative proportion of these states changes across the night, reflecting shifts in neural priorities as sleep progresses. Non-REM sleep constitutes the majority of total sleep time and is characterized by progressively slower and more synchronized brain activity. REM sleep, by contrast, is marked by desynchronized cortical activity, vivid dreaming, and profound muscle atonia. Together, these states form a dynamic system in which different neural processes dominate at different times. Electroencephalography, or EEG, has been central to understanding the brain's activity during sleep. EEG recordings reveal that sleep is associated with distinct patterns of electrical oscillations, commonly referred to as brain waves. These oscillations reflect the coordinated activity of large populations of neurons and provide insight into the functional state of the brain. During wakefulness, EEG signals are dominated by low amplitude, high-frequency beta and gamma waves, reflecting continuous sensory processing, attention, and executive control. As sleep begins, this pattern changes gradually. Alpha activity diminishes, sensory responsiveness declines, and slower oscillations emerge. These changes signal a shift from externally oriented cognition to internally driven neural processing. Non-REM sleep is subdivided into 3 stages, each defined by characteristic EEG patterns and neural functions. The first stage represents the transition from wakefulness to sleep. During this stage, theta waves begin to replace alpha activity, and awareness of the external environment decreases. Although consciousness is reduced, the brain remains responsive to salient stimuli, and brief awakenings are common. The second stage of non-REM sleep is defined by the appearance of sleep spindles and K-complexes. Sleep spindles are short bursts of rhythmic activity in the sigma frequency range, while K-complexes are large, slow waveforms that reflect cortical responses to internal or external stimuli. These features are not merely markers of sleep depth. They play an active role in suppressing sensory input and stabilizing the sleeping state. Sleep spindles, in particular, have been closely linked to learning and memory. Their occurrence correlates with improvements in both declarative and procedural memory tasks, suggesting that they facilitate the transfer of information within neural networks. At the same time, K-complexes appear to protect sleep by preventing minor disturbances from triggering awakenings. As sleep deepens further, the brain enters the third stage of non-REM sleep, commonly referred to as slow-wave sleep. This stage is dominated by delta waves, which are high amplitude, low frequency oscillations reflecting widespread neuronal synchronization. During slow wave sleep, large populations of cortical neurons alternate between periods of activity and silence in a highly coordinated manner. This synchronized activity represents a fundamental shift in how the brain operates. Rather than processing information from the external environment, the brain focuses on internal maintenance and reorganization. Metabolic demands decrease, cerebral blood flow is redistributed, and neurochemical conditions favor restoration and plasticity. Slow wave sleep is particularly important for physical recovery and brain health. Growth hormone secretion peaks during this stage, supporting tissue repair and immune function. At the neural level, slow wave activity is associated with synaptic downscaling, a process through which the brain weakens less important synaptic connections while preserving those that are most relevant. This mechanism helps maintain network efficiency and prevents synaptic saturation. In addition to synaptic regulation, slow wave sleep plays a central role in memory consolidation. During wakefulness, new information is rapidly encoded by the hippocampus, a medial temporal lobe structure specialized for forming episodic memories. However, hippocampal storage capacity is limited, and newly formed memory traces are initially fragile. Long-term storage requires their gradual integration into distributed cortical networks. During slow-wave sleep, the hippocampus repeatedly reactivates patterns of neural activity that were present during learning. These reactivations occur in coordination with cortical slow waves and thalamic sleep spindles, creating a temporal framework that supports the transfer of information from hippocampal to neocortical regions. Through this process, memories become more stable, more generalized, and less dependent on the hippocampus. While non-REM sleep is dominated by slow synchronized activity, REM sleep represents a strikingly different neural state. REM sleep is characterized by low amplitude, high frequency EEG activity resembling that of wakefulness. Despite this apparent cortical activation, behavioral responsiveness remains minimal and voluntary muscle activity is almost completely suppressed. The defining features of REM sleep include rapid eye movements, vivid dreaming, and profound muscle atonia. These features are generated by specialized circuits within the brainstem that regulate arousal, motor inhibition, and autonomic function. During REM sleep, cholinergic neurons in the pons become highly active, while monoaminergic systems that promote wakefulness are strongly inhibited. This neurochemical environment supports intense internal processing while preventing physical enactment of dreams. Brain imaging studies have shown that REM sleep is associated with increased activity in limbic and paralimbic regions, including the amygdala, hippocampus, and anterior cingulate cortex. These areas are involved in emotion, memory, and motivational processing. In contrast, activity in the dorsolateral prefrontal cortex, a region critical for executive control and logical reasoning, is significantly reduced. This pattern of activation helps explain the distinctive characteristics of dreams. Dream narratives often involve strong emotional content, fragmented logic, and altered perceptions of time and space. The reduced influence of prefrontal control allows internally generated associations to unfold without the constraints of rational evaluation. REM sleep appears to play a particularly important role in emotional regulation. During this stage, emotional memories are reprocessed in a neurochemical context that minimizes stress-related signaling. Levels of noradrenaline, a neurotransmitter associated with arousal and threat detection, are markedly reduced during REM sleep. This reduction allows the brain to revisit emotionally charged experiences without triggering the full physiological stress response associated with wakefulness. Through repeated cycles of REM sleep, emotional memories may be gradually decoupled from their original affective intensity. This process helps explain why the emotional impact of experiences often diminishes over time, and why disruptions in REM sleep are closely linked to mood disorders and trauma-related conditions. Beyond memory and emotion, sleep supports a critical housekeeping function that has only recently been fully appreciated. The brain lacks a conventional lymphatic system, raising the question of how metabolic waste products are removed from neural tissue. Research has shown that the brain relies on a specialized clearance pathway known as the glymphatic system. The glymphatic system facilitates the exchange of cerebrospinal fluid and interstitial fluid, allowing metabolic byproducts to be flushed out of the brain. This system is most active during sleep, particularly during slow-wave sleep. As neuronal activity decreases and cells shrink slightly, the spaces between neurons expand, enabling more efficient fluid flow. Among the substances cleared through this process are beta-amyloid and tau proteins, which are associated with neurodegenerative diseases. Impaired sleep reduces glymphatic clearance, leading to the accumulation of these proteins over time. This mechanism provides a biological explanation for the strong association between chronic sleep disruption and increased risk of neurodegenerative disorders. Sleep also plays a fundamental role in maintaining neural plasticity. Plasticity refers to the brain's ability to change its structure and function in response to experience. While learning during wakefulness strengthens specific synaptic connections, sleep allows the brain to recalibrate overall synaptic strength, preserving signal-to-noise balance and preventing runaway excitation. Without adequate sleep, synaptic regulation becomes impaired, neural networks become inefficient, cognitive flexibility declines, and the brain's capacity to adapt to new information is reduced. These effects can be observed after even short periods of sleep deprivation and become more pronounced with chronic sleep loss. Sleep deprivation has widespread consequences for brain function. Cognitive performance declines, attention becomes unstable, and decision-making processes are compromised. Neuroimaging studies show reduced activity in the prefrontal cortex, accompanied by heightened reactivity in emotional circuits. This imbalance leads to impaired judgment, increased impulsivity, and exaggerated emotional responses. At the cellular level, sleep deprivation alters gene expression related to synaptic function, energy metabolism, and stress response. Over time, these changes contribute to increased vulnerability to psychiatric disorders, metabolic dysregulation, and neurodegenerative disease. Despite its critical importance, sleep is often sacrificed in modern society. Artificial lighting, irregular schedules, and constant cognitive stimulation disrupt natural sleep patterns. From a neuroscientific standpoint, this disruption represents a fundamental mismatch between evolved brain mechanisms and contemporary environmental demands. Understanding what the brain does during sleep reveals that sleep is not a passive state to be minimized, but an active biological necessity that supports nearly every aspect of neural function. Sleep enables the brain to stabilize memories, regulate emotions, clear metabolic waste, and preserve the capacity for adaptation. Without sleep, these processes fail, and the integrity of the brain gradually deteriorates.
Sleep, therefore, is not merely a period of rest. It is a distinct and essential mode of brain operation, without which sustained cognitive and emotional function would not be possible.
Sleep also plays a critical role in the regulation of consciousness. Consciousness is not an all-or-nothing phenomenon, but exists across a spectrum of states defined by patterns of neural connectivity, information integration, and responsiveness. Sleep represents a unique configuration within this spectrum, characterized by reduced external awareness but preserved internal processing. During wakefulness, consciousness is dominated by sensory input and goal-directed cognition. Information flows predominantly from sensory systems toward associative and executive networks. During sleep, particularly during non-REM sleep, this flow is largely reversed. External input is actively suppressed, and internally generated activity becomes the primary driver of neural dynamics. This shift is mediated by changes in thalamocortical connectivity. The thalamus, which serves as a relay station for sensory information, alters its firing mode during sleep. Instead of transmitting sensory signals to the cortex, thalamic neurons enter a burst firing mode that effectively blocks external information. This gating mechanism is essential for maintaining sleep continuity and enabling internal neural processes to dominate. Despite this sensory disconnection, the brain does not lose its capacity for information processing. Instead, it operates under a different set of constraints. Large-scale brain networks reorganize, and patterns of functional connectivity change. Networks associated with self-referential thought, memory retrieval, and emotional processing remain active, while networks involved in executive control and attentional monitoring are downregulated. This reorganization allows the brain to process information without the constant demands of the external environment. It also creates the conditions necessary for phenomena such as dreaming, memory replay, and emotional recalibration. Lucid dreaming provides a particularly informative window into the relationship between sleep and consciousness. During lucid dreams, individuals become aware that they are dreaming while remaining in the sleep state. Neuroimaging studies suggest that lucid dreaming involves partial reactivation of prefrontal regions typically suppressed during REM sleep. This partial restoration of executive function allows reflective awareness to emerge without full awakening. Lucid dreaming demonstrates that elements of conscious awareness can coexist with the neural architecture of sleep. This finding challenges simplistic models that equate consciousness strictly with wakefulness and suggests that consciousness is a flexible property arising from specific patterns of neural interaction rather than a single brain state. Beyond individual experiences, sleep must also be understood from an evolutionary perspective. Sleep is observed across species with vastly different nervous systems, from insects to mammals. While the structure and duration of sleep vary widely, the presence of sleep-like states across phylogeny indicates that sleep confers a strong adaptive advantage. One evolutionary hypothesis proposes that sleep evolved as a means of energy conservation. While reduced activity during sleep does lower metabolic demand, this explanation alone is insufficient. Many animals sleep in environments where inactivity increases vulnerability to predation, suggesting that the benefits of sleep must outweigh its risks. Another hypothesis emphasizes the role of sleep in neural maintenance. Brains are energetically expensive organs, and neural activity generates metabolic byproducts that must be cleared to prevent cellular damage. Sleep provides a protected temporal window during which restorative processes can occur without interference from sensory processing and motor activity. From this perspective, sleep can be viewed as an essential maintenance state that preserves the long-term functionality of neural circuits. Species with more complex brains tend to exhibit more structured sleep patterns, supporting the idea that sleep complexity scales with neural complexity. Sleep architecture also changes systematically across the human lifespan, reflecting shifts in neural development and functional priorities. Infants spend a much larger proportion of sleep in REM sleep compared to adults. This high REM density is thought to support rapid brain development, synaptogenesis, and the establishment of neural circuits. As development progresses, the proportion of REM sleep decreases, while non-REM sleep becomes more prominent. Slow-wave sleep is particularly abundant during childhood and adolescence, coinciding with periods of intense synaptic pruning and cortical maturation. During adulthood, slow-wave sleep gradually declines, reflecting age-related changes in neural plasticity and homeostatic regulation. In older adults, sleep becomes more fragmented, and the ability to generate deep slow-wave sleep diminishes. These changes are associated with impairments in memory consolidation and increased vulnerability to neurodegenerative processes. While aging-related sleep changes are partly unavoidable, maintaining sufficient sleep quality remains critical for preserving cognitive function. Sleep disorders provide further insight into the functions of sleep by revealing what happens when specific sleep processes are disrupted. Insomnia, characterized by difficulty initiating or maintaining sleep, is associated with hyperarousal of neural systems that normally downregulate during sleep. Neuroimaging studies show increased activity in wake-promoting regions even during attempted sleep, suggesting a failure of normal inhibitory mechanisms. Sleep apnea, which involves repeated interruptions of breathing during sleep, leads to chronic fragmentation of sleep architecture. Although total sleep time may appear adequate, the repeated arousals prevent sustained engagement in slow-wave and REM sleep. This disruption impairs memory, attention, and emotional regulation, illustrating the importance of sleep continuity in addition to sleep duration. Narcolepsy, a disorder characterized by excessive daytime sleepiness and abnormal intrusion of REM sleep into wakefulness, highlights the role of sleep-wake boundaries in maintaining stable consciousness. Narcolepsy is associated with loss of hypocretin-producing neurons in the hypothalamus, which normally help stabilize arousal states. The resulting instability demonstrates that precise neurochemical regulation is required to maintain distinct sleep and wake states. Circadian rhythms further modulate sleep and brain function. The circadian system, governed by the suprachiasmatic nucleus in the hypothalamus, synchronizes sleep timing with environmental light-dark cycles. This synchronization ensures that sleep occurs at biologically optimal times, aligning restorative processes with predictable periods of reduced environmental demand. Disruption of circadian rhythms, such as through shift work or irregular sleep schedules, impairs sleep quality even when total sleep duration is preserved. Misalignment between circadian signals and sleep timing leads to reduced slow wave activity, altered REM distribution, and impaired metabolic and cognitive function. These effects underscore the importance of sleep timing as well as sleep quantity. Taken together, these findings indicate that sleep is not a single-purpose state, but a multifaceted biological process supporting neural integrity at multiple levels. Sleep enables the brain to alternate between different modes of operation, each optimized for specific functions that cannot be efficiently performed during wakefulness. The brain during sleep is engaged in active computation, reorganization, and maintenance. It consolidates memories, recalibrates emotional responses, clears metabolic waste, preserves synaptic balance, and maintains the stability of conscious experience. These processes are interdependent, forming an integrated system that sustains cognitive and emotional health. The absence or disruption of sleep compromises this system, leading to cascading effects that extend far beyond transient fatigue. Over time, insufficient sleep erodes the brain's capacity to learn, adapt, and regulate internal states. The consequences of chronic sleep disruption accumulate gradually, often remaining unnoticed until significant dysfunction emerges. From a neuroscientific perspective, sleep is therefore not optional. It is a fundamental requirement for the continued operation of the brain as a complex adaptive system. The brain does not sleep in order to shut down. It sleeps in order to perform functions essential to its own survival. One of the most important insights emerging from sleep research is that the brain does not simply alternate between being active and inactive. Instead, it transitions between distinct operational modes, each defined by specific patterns of neural communication, chemical signaling, and functional priorities. Sleep represents a set of highly regulated brain states in which neural activity is redistributed rather than reduced. During wakefulness, neural resources are allocated toward processing unpredictable sensory input, coordinating motor actions, and maintaining goal-directed behavior. These demands require continuous engagement of executive networks and rapid information exchange across distributed brain regions. Sleep removes these external pressures, allowing the brain to reallocate resources toward internal optimization. A central component of this optimization is synaptic regulation. Throughout waking life, synapses are constantly modified in response to experience. Learning strengthens certain connections, while others weaken or remain unchanged. Without a counterbalancing process, this continual synaptic strengthening would lead to excessive energy consumption, reduced signal specificity, and impaired network stability. Sleep provides the conditions necessary for restoring synaptic balance. According to the synaptic homeostasis hypothesis, slow-wave activity during non-REM sleep promotes a global downscaling of synaptic strength. This downscaling does not erase learning. Instead, it preserves relative differences between synapses while reducing overall synaptic load. As a result, essential connections are maintained, while redundant or weakly relevant connections are selectively pruned. This process improves signal-to-noise ratios within neural networks and enhances learning capacity upon awakening. Experimental evidence shows that individuals deprived of sleep exhibit reduced ability to encode new information, consistent with the idea that synaptic saturation limits further plasticity. By contrast, sleep restores the brain's readiness for learning by recalibrating synaptic weights. Neurochemical changes during sleep play a critical role in shaping these processes. Levels of neuromodulators such as acetylcholine, noradrenaline, serotonin, and dopamine fluctuate systematically across sleep stages. These changes alter neuronal excitability, plasticity mechanisms, and network dynamics. During non-REM sleep, reduced acetylcholine levels favor communication between hippocampus and neocortex, facilitating memory consolidation. During REM sleep, elevated acetylcholine combined with suppressed monoaminergic activity creates conditions that support associative processing and emotional integration. These distinct chemical environments enable different forms of neural computation to occur at different times during the night. Sleep also influences gene expression within the brain. Studies examining transcriptomic changes reveal that sleep and wakefulness are associated with distinct patterns of gene activation. Genes involved in synaptic plasticity, membrane maintenance, and cellular repair are preferentially expressed during sleep, while genes associated with stress response and energy metabolism are more active during wakefulness. These molecular shifts suggest that sleep supports long-term brain health by coordinating cellular maintenance with network-level reorganization. Without sufficient sleep, this coordination breaks down, leading to cumulative cellular stress and impaired neural resilience. Another critical function of sleep involves the regulation of immune signaling within the brain. Neuroinflammatory processes are tightly linked to sleep-wake cycles. Acute sleep loss increases markers of inflammation both centrally and peripherally. Chronic sleep disruption is associated with sustained inflammatory activation, which can negatively affect synaptic function and neuronal survival. By contrast, sleep promotes anti-inflammatory signaling and supports immune surveillance mechanisms that protect neural tissue. This relationship provides further insight into why sleep disturbances are associated with increased risk of neurological and psychiatric disorders. Sleep's role in emotional regulation extends beyond memory reprocessing. By modulating the sensitivity of emotional circuits, sleep helps maintain stable, effective responses. When sleep is disrupted, the balance between prefrontal regulatory systems and subcortical emotional centers becomes unstable. This instability manifests as heightened emotional reactivity, reduced impulse control, and impaired stress tolerance. Neuroimaging studies demonstrate that even a single night of sleep deprivation can amplify amygdala responses to negative stimuli while weakening functional connectivity between the amygdala and prefrontal cortex. These changes mirror patterns observed in mood and anxiety disorders, underscoring the importance of sleep for emotional stability. Sleep also supports social cognition and decision-making. Complex social judgments require integration of emotional cues, memory, and executive control. Sleep deprivation disrupts this integration, leading to biased judgments, reduced empathy, and impaired moral reasoning. These effects highlight sleep's contribution not only to individual cognition, but also to social functioning. The relationship between sleep and mental health is bidirectional. Sleep disturbances increase vulnerability to psychiatric disorders, while psychiatric conditions often disrupt sleep architecture. Depression, anxiety disorders, bipolar disorder, and schizophrenia are all associated with characteristic alterations in sleep patterns. Understanding these alterations provides insight into shared neurobiological mechanisms underlying sleep and psychopathology. For example, reduced slow-wave sleep and altered REM sleep timing are common features of major depressive disorder. These changes may reflect dysregulation of monoaminergic systems and impaired emotional memory processing. Therapeutic interventions that normalize sleep patterns often produce improvements in mood, suggesting that sleep modulation can influence core disease processes. Sleep's influence on cognitive aging further illustrates its importance. Age-related declines in memory and executive function are exacerbated by sleep disruption. Older adults who maintain better sleep quality show slower rates of cognitive decline and reduced risk of dementia. While sleep alone cannot prevent neurodegeneration, it appears to modulate the brain's capacity to compensate for age-related changes. At the systems level, sleep enables coordination across distributed neural networks. Brain function depends on dynamic interactions among specialized regions rather than isolated activity within individual areas. Sleep alters the strength and configuration of these interactions, promoting network flexibility and resilience. Functional connectivity studies reveal that sleep reorganizes network topology, reducing rigid patterns established during wakefulness and enabling alternative configurations to emerge. This flexibility may underlie the brain's capacity for insight, creativity, and adaptive problem solving. Despite the growing understanding of sleep's functions, many aspects of sleep neurobiology remain incompletely understood. The precise mechanisms linking specific oscillatory patterns to distinct cognitive outcomes are still being investigated. The relationship between subjective experience, such as dream content, and objective neural activity remains an active area of research. Nevertheless, the convergence of evidence from electrophysiology, neuroimaging, molecular biology, and behavioral studies supports a unified conclusion. Sleep is a fundamental biological process that supports the brain's structural integrity, functional organization, and adaptive capacity. The brain does not use sleep to disengage from activity. It uses sleep to perform operations that are incompatible with wakefulness. These operations ensure that neural systems remain efficient, flexible, and capable of supporting conscious experience over time. In this sense, sleep represents an alternative mode of brain function, optimized for maintenance, integration, and long-term stability. The transition between wakefulness and sleep is not a shutdown, but a shift in priorities. The brain continues to work, guided by different rules and serving different goals. Understanding what the brain does during sleep provides a deeper appreciation of why sleep occupies such a large portion of human life. Sleep is not time lost. It is time invested in preserving the brain's ability to function. As research continues to clarify the mechanisms underlying sleep, one conclusion has become increasingly difficult to ignore. Sleep is not a secondary biological process that supports waking life from the background. It is a core operational state of the brain, essential for maintaining the conditions under which waking cognition can exist at all. The functions carried out during sleep are not optional optimizations. They are foundational requirements for neural stability. Memory systems rely on sleep to prevent overload and distortion. Emotional circuits depend on sleep to recalibrate sensitivity and maintain balance. Cellular maintenance mechanisms require sleep to prevent the accumulation of metabolic stress. Network-level organization depends on sleep to preserve flexibility and efficiency. When sleep is reduced or disrupted, these systems do not simply pause, they degrade. The effects may not be immediately visible, but they accumulate over time, altering cognitive performance, emotional regulation, and neurological resilience. Sleep loss compromises the brain not by causing sudden failure, but by slowly eroding the processes that sustain its function. The brain's reliance on sleep reflects fundamental constraints of biological computation. Neural systems cannot simultaneously process continuous external input, support adaptive learning, maintain synaptic balance, and perform large-scale cellular maintenance. Sleep resolves this constraint by separating these functions across time. Wakefulness prioritizes interaction with the environment. Sleep prioritizes internal regulation. This temporal separation allows the brain to operate efficiently without interference. During sleep, neural activity is reorganized rather than silenced. Patterns of connectivity are reshaped, unnecessary associations are weakened, and essential structures are reinforced. The result is not a passive recovery, but an active reconfiguration that prepares the brain for future demands. From this perspective, sleep can be understood as a complementary state to wakefulness. Each depends on the other. Wakefulness generates experience and information. Sleep integrates, stabilizes, and regulates the neural consequences of that experience. Neither state is sufficient on its own. The persistence of sleep across evolution underscores its importance. Despite the risks associated with reduced awareness and immobility, sleep has been conserved across species with vastly different ecological pressures. This conservation suggests that the costs of sleep are outweighed by the catastrophic consequences of its absence. In humans, the complexity of sleep architecture mirrors the complexity of cognitive function. The coordination of non-REM and REM sleep, the precise timing of neural oscillations, and the modulation of neurochemical systems reflect an evolved solution to the demands placed on an adaptable, learning-capable brain. Understanding sleep as an active brain state challenges cultural assumptions that equate productivity with wakefulness alone. From a neuroscientific standpoint, sustained cognitive performance depends not on extending waking hours, but on preserving the integrity of sleep-dependent processes. Sleep does not compete with waking life, it enables it. The brain does not sleep in order to escape activity, it sleeps in order to continue functioning.